All right, thank you for tuning into this virtual presentation. I regret that I was unable to attend Glasgow due to a couple of important personal events, a wedding of a very close friend and residency graduation that are on the same weekend. So I'm going to talk about multimodal parcellation of the human cerebral cortex and three main topics. First, a primer on neuroimaging HCP style, how to acquire, preprocess, and analyze your data to enable accurate parcellation. Then we'll talk about a multimodal gradient-based semi-automated neuroanatomical cortical parcellation, the HCP's multimodal cortical parcellation version 1.0. And then about parcellation of individual subjects, basically how we're going to use machine learning to enable automated multimodal cortical parcellation of even atypical subjects. So first, the primer on HCP style neuroimaging. So what do I mean by that? Well, collecting as much high quality multimodal imaging data on as many subjects as possible, using MRI acquisitions with adequate spatial resolution to map the brain structures that you're studying, using care during data preprocessing to remove distortions in motion without causing blurring, representing MRI data according to the natural geometry of the brain structures, meaning surfaces for the cerebral cortex, aligning cortical areas, not cortical folds across subjects, using a data-driven, functionally and structurally sensible parcellation, and routine sharing of published results to enable direct comparisons across studies. So what kind of MRI data do we need for multimodal parcellation? Well, as you've heard in other talks in this session, parcellations have been defined based on architecture, function, connectivity, and topography. And we can measure architecture with T1 weighted and T2 weighted images with MRI as myelin maps or cortical thickness maps. And we can use fMRI to measure the other three: function, connectivity, and topography. And so the HCP acquired and analyzed these data on over 1,000 subjects at 3T and just under 200 subjects at 7T. So when thinking about acquiring HCP-style MRI data, we really need to think about how neuroanatomy drives the spatial resolution choices. So this is a group average of mean cortical thickness of the cortical thickness, and mean cortical thickness we can see in this histogram is 2.6 millimeters. We'll call fMRI resolutions that are coarser than 2.6 millimeters, low resolution fMRI. High resolution fMRI is those resolutions that are higher than 2.6 millimeters. And we'll note that the minimum thickness of cortex is about 1.6 millimeters. So in the 3T data set, we were at 2 millimeter isotropic, getting one frame every 0.72 seconds. And at 7 Tesla, we were at 1.6 millimeter isotropic with one frame every second. Other things to think about for our structural MRI, we really want to be at least 0.8 millimeter isotropic or better. That gives us at least 2 voxels for even the thinnest cortex to make sure we get accurate surface meshes and segmentations. At 3 Tesla on Siemens, the MP range and T2 space work very well with the 32 channel head coil. Functional MRI, we want our resolution to be better than 2.6 millimeter isotropic. For example, 2 millimeter isotropic, we want to image fast, less than a second for denoising purposes. And we want to get plenty of data, at least 30 minutes of data and more is better. We've tested these methods on resting state, but other kinds of fMRI are likely to work as well. And to hit these targets of temporal and spatial resolution really require a multi-band EPI sequence. Remember to save the single band reference image, auxiliary images to get at the same time, some kind of a field map, and we prefer 2 phase reverse spin echo images that match the fMRI data in terms of their geometry and distortion. We can use these to correct for B0 distortion, B1 image intensity and homogeneity, or even estimate B1 plus transmit field and homogeneity. And all of the analyses I'm going to talk about today require just these 4 images. And one thing we've learned more recently is it really helps to use Siemens pre-scan normalize or the equivalent on all of these. So we would recommend that. What about careful pre-processing? What do we need to do? Well, we want to remove MR-based image distortions so that all images represent the physical space of the subject. Align images across and within modalities, removing the spatial effects of motion. Make geometric models of the brain's major structures, cortical surface meshes, and a subcortical segmentation. Avoid blurring the data with multiple interpolations or smoothing, overt smoothing. Avoid inaccurate alignment of brain areas across subjects. So how should we represent the data to maximize our alignment accuracy across subjects? Well, the HCP developed a new analysis framework for accurate alignment of brain areas across subjects. We're going to consider gray matter structures according to the geometric model best suited for each, surfaces for the sheet-like cerebral cortex, and volumes for globular subcortical nuclei. And that's illustrated as the SIFTY gray ordinate standard space, which we see here has 30,000 left cortical surface vertices, 30,000 right cortical surface vertices, and 30,000 voxels across 10 subcortical structures, giving us a total of 90,000 gray ordinates. So we're going to register individuals' cortical data using nonlinear surface registration and their subcortical data using nonlinear volume registration. And SIFTY gray ordinance-based imaging analysis really greatly reduces the uncertainty in spatial localization that's found in traditional brain imaging studies. Just as an example of this, we can take area M2 and see what happens if we align it with a volume-based registration. Doesn't work very well. We've got sort of two hotspots of probability for area MT and kind of just a blurry mess here. Our overlap is less than 50%. A folding-based registration does do better. Our overlap's over 50%. We've got a bigger single hotspot of high probability. We've still got individual subjects whose cortical areas are not aligned to that hotspot. Turns out an aerial feature-based surface registration does quite a bit better, gets us to 100% overlap. And by aerial features, I mean things like architecture, function, connectivity, topography, or the areas themselves. So there are a number of tools that allow you to do neuroimaging HCP style in terms of acquisition protocols and pulse sequences. A couple of websites here to get those. Software preprocessing pipelines and binaries, the HCP's minimal preprocessing pipelines, which include MSM-all aerial feature-based alignment. The MSM software itself is available here. For visualizing SIFTY gray ordinates and doing analyses, Connectome Workbench and Workbench Command are really important tools, but there's also some capabilities for both FSL and MATLAB and other things now, Python and so on. And then in terms of the data, fully preprocessed and aligned HCP multimodal data in over 1,000 3T subjects and just under 270 subjects. I regret I'm not able to take your questions live today, but I'm more than happy to answer them on the HCP users mailing list. I'm on that list every day answering questions and happy to take any questions that you have. All right, so let's talk about a multimodal gradient-based semi-automated neuroanatomical parcellation, basically the HCP's multimodal parcellation version 1.0. Well, we could take a step back, and other folks in the session have likely also discussed this question. Why parcellate the cerebral cortex? Well, it helps us to know where we are in the brain if we have a map, and it helps communication between investigators and aids comparisons across studies. If we can agree on an area's boundaries, we can study its properties in detail. And an example I like to give about this is, Serena et al.'s science paper looking at early visual areas. He was able to recognize area V1, V2, V3, V4, very similar to what we do today, even more than 27 now years ago. Parcellation reduces data size and complexity while enhancing statistical sensitivity and power. You can think of it as a neuroanatomically informed smoothing method, and it avoids many of the methodological imprecisions that have been endemic in brain imaging, such as M&I coordinate tables and for studies that are interested in area level effects. So what do we want in a cortical parcellation? Well, I think it should ideally have 4 qualities. Ideally, it's based on hundreds of precisely aligned subjects so that it represents the typical areal arrangement of a population. Ideally, it has complementary and converging evidence from multiple modalities across the whole cerebral cortex, reflects the terminology used in the neuroanatomical literature, and ideally, we'd be able to take this group parcellation and be able to automatically replicate it in individual subjects based on multimodal aerial fingerprints. And so the HCP's cortical parcellation version 1.0 has all 4 of these qualities. So what do I mean by hundreds of well-aligned subjects? Well, here's a myelin map where our myelins high myelins are in the reds, yellows, and greens, and low myelins are the blacks, purples, and blues. So this is a group of 210 subjects that we're going to call 210P and use throughout the data set. Here's an independent group of 210 subjects. We're going to call it 210V for validation. And if we compare these maps, they look really similar, even down to the fine spatial details, like this little dip in myelin content between the face and upper extremity somatosensory cortex. It shows up in both data sets. And this is not your typical blurry group average here. And you can do the same thing with task fMRI. So we haven't smoothed the task fMRI data. Most people smooth task fMRI data, but you don't have to. And if you average it across a bunch of subjects, you can even find highly reproducible little activations like this one in area 55B. And so these correlations between these group averages are really high. They're 0.9, 0.98 for myelin, for a task, which I'm showing here, or resting state that I'm not showing. So how do we turn this multimodal data into a cortical parcellation? Well, most extant parcellations, including those that you've seen elsewhere in the session, were generated with only one of the modalities of architecture, function, connectivity, and topography, but the HCP measured all four. And so we can use gradients or derivatives with respect to space as an objective measure of where each modality is changing rapidly. And these are potential areal boundaries, and they provide a common space in which we can compare across modalities. What makes a gradient convincing is an areal boundary. Well, number one is if we've got agreement in location between two or more independent modalities, but statistically robust and significant evidence of multimodal differences across the boundary is also very important. If it's present in both hemispheres, that's an internal replication, not known, associated with a known imaging artifact. If we've got prior literature evidence, sort of in a Bayesian sense. And then once we have the set of boundaries that we think are the ones that are present in the cerebral cortex, we can relate those to the existing literature to identify previously described areas or describe new ones. So how does this work in individual modalities? Well, we consider myelin maps again. And now we're going to focus on where the myelin content changes. So the myelin gradient here, the reds, yellows, oranges, and greens are showing where the myelin gradient, myelin content is changing rapidly. And the local maxima of gradient is the most likely location of an potential areal border. And some of these transitions are larger than others, but it's those transitions that occur in multiple modalities that are going to be most interesting as areal border candidates. So we can do cortical thickness as another approach that gives us architectural information. We can take a gradient of cortical thickness just like a myelin map and see, this strong transition between thicker motor cortex and thinner somatosensory cortex right in the central sulcus here. In this case, we've regressed out curvature to reduce the effects of folding on thickness and focus on the effects of cortical areas. You can do it in task fMRI as well. So the positive areas have more activity while listening to a story, whereas the negative areas have more activity during resting. We can take a gradient of a task fMRI effect size map or beta map, just like we did of an architectural measure. Functional connectivity we can make gradients with. So here's a seed location on this white circle here, and areas that are functionally connected are the positive areas, the reds, yellows, oranges, and greens. We can make a gradient of where functional connectivity patterns change as we step across the cortex. So here we step across a strong gradient and we could see a strong change in functional connectivity patterns, step across another one, and we've got another change back to something more similar to what we had at the beginning. We focus on this particular pattern in between here and compare it to a task activation map. We can see they're very similar, illustrating that areas that are activate together are often functionally connected. What about topography? Not enough time to discuss this in detail today, but it's possible to map out visuotopy with retinotopic tasks or with resting state fMRI. And in either case, topography enables mapping of visual areas, whereas connectivity would tend to split visual areas according to topographic gradients. We can also use topography to map out subareas of somatosensory and motor cortex. So let's put all this together for one cortical area. How do we do multimodal parcellation? I'm going to focus on this strip of lightly myelinated cortex right here. We've drawn these white dots along the myelin gradients, defining the most likely areal boundaries. Then we look at another modality, listening to a story, and we find unique task activation within this particular region and not in the neighbors. We've got strong task gradients lining up with the myelin gradients. We can look in functional connectivity now and put a seed in another location within the language network here. And we've got specific connectivity to this particular area, but not its neighbors. But if we put a seed in a different functional network here in LIPV here, we see selective connectivity to the I fields and not to the cortical area of interest. So we've got functional connectivity gradients that line up with the myelin and the task gradients, multiple independent modalities, architecture, function, and connectivity, all agreeing on a cortical area. And so the last step is to identify this area with respect to the literature. It turns out that Adolf Hopf made some nice hand-drawn surface maps of myelin density where we have our two I fields on either side, just like we see here in our in vivo myelin maps. Got our motor cortex behind this location, just like we see here. And he called this location of slightly lighter myelin area 55B. And so we can take that going forward. So the multimodal parcellation was constructed from the 210P or parcellation group of subjects using borders defined on gradients and group average architecture, function, connectivity, and topography. We use a semi-automated approach where the neuroanatomists chose the useful modalities for each areal boundary and drew an initial border, and then an algorithm optimized the boundary to best follow the multimodal gradients. And these areas were defined with reference to the prior neuroanatomical literature, keeping the same names when possible. So we went into this exercise with several predictions based on monkey parcellation and partial human parcellation. We thought there'd be somewhere between 150 to 200 cortical areas per hemisphere, wide variability in areal size and shape across the cortex, examples of interareal heterogeneity, such as sensory motor topographies. And here are the borders. We found 180 areas per cortical hemisphere. 97 were new and 83 were existing. There's wide variability in areal shape and size, anywhere from long, thin strips in sensory motor cortex matching known cortical areas in this region to horseshoe-shaped strips in visual cortex, again, matching known cortical areas. And so, and every shape in between. And so we were able to, with this approach, actually recover the cortical areas that had already been well defined in humans by prior techniques, along with many others. And some of these areas do contain topographic sub-areas, such as the sensory motor areas that are, those boundaries are not shown here. So we can colorize the parcellation where we have reds for auditory, greens for sensory motor, and blues for visual, darker colors for default mode or task negative, and brighter colors for anti-default mode or task positive networks, with mixed colors in between for areas like LIPV here that have both sensory motor and visual inputs. Now, we've done all this in the 210P group of subjects, but in an independent group of subjects, are the areal features really different across these parcel boundaries? So we're going to take categories of cortical thickness, myelin content, task activation, and resting state connectivity. And we're going to put up some pretty high thresholds. We're going to expect robust effect sizes, a Cohen's d of greater than 1 and a Bonferroni corrected p-value for a two-tailed test for over 1,000 border pairs. So the red borders are those where all 4 categories pass these thresholds, and that's about a quarter. About half or orange had 3 categories. Another quarter had 2 categories, and really only a minority had only 1 or no categories. And these effects were driven actually mainly by the effect size threshold of 1, the Cohen's d threshold of 1, as all borders had at least 2 modalities that were significant to the Bonferroni corrected p-value. And so this method can also be used to show which modalities were useful for distinguishing each border. Cortical thickness showed some borders, myelin showed more, task showed even more, and resting state showed the most. Okay, so now we have a parcellation. What can we do with it? We've got our areal boundaries overlaid on a myelin map. And we have about 50,000 data points here, almost 60,000 data points that we can see. But if we average the myelin content within each brain area, we can get that down to only 360 data points. And if I flip between the two, you really don't see that much difference in the pattern between the dense analysis and the parcellated analysis. But we've gotten 2 orders of magnitude decrease in our data size, our number of statistical tests, along with increasing our signal to noise ratio. So we can do parcellated analyses for functional connectivity here with a seed in the left inferior parietal cortex, looking at default mode. For full correlation, partial correlation is a sparser matrix, but still shows a nice default mode network. We can do this with task fMRI as well. Here's a working memory task. Here's listening to a story. And, particularly if we look at the story contrast and we've got our parcellated analysis down here and a traditional surface-based dense analysis up here, we can see that the statistics are quite a bit higher, scaled the same for the parcellated analysis versus the dense. And so there really is a benefit to parcellating a time series before fitting a GLM versus, for example, just averaging Z statistics within a parcel. And if those approaches were equivalent, the blue dots would be along this red line here, but they're steeper than the red line, indicating that parcellating first and fitting the GLM second is the better approach. So to summarize this section, 180 cortical areas in each hemisphere, which was within our expected range with 97 new areas and 83 existing areas. They vary widely in size and shape, and some areas have topographic heterogeneity. We can do parcellated analyses with most modalities, including architecture, function, or connectivity. And there's minimal loss of detail at the areal level with a good parcellation. The HCP's multimodal parcellation is available at this website. Balsa is a new database for sharing HCP-style published results. And it's really handy because you can actually make and annotate multi-panel figures inside Connectome Workbench. No need to use PowerPoint or an image editor. You can save these figures as scenes and come back to them and modify them later. And when you're ready, export an image for your paper, and then you can take that scene and upload it to Balsa, and you've shared all of the data necessary to make the figure. Again, any questions, please let us know on the HCP users list. I'm on that list every day and happy to answer any of your questions. I apologize that I can't take them live today. All right, so let's close with a discussion of parcellation of individual subjects and how we can use machine learning to get there. So first, let's motivate the problem, considering again area 55B and some of the atypical patterns that can be seen. So the typical relationship between area 55B and the upper limb sensory motor subregion looks like this. But some subjects have an atypical relationship where 55B is located more superiorly relative to this large region here. And so aerial feature-based registration preferentially is going to align this larger feature of the upper limb sensory motor subregion, and that causes a shift, upward shift of area 55B. Some real examples. So the group average location of 55B is outlined in black and white here. And these are group average language network with resting state, language task activation, myelin map, and the upper limb sensory motor subregion. A typical subject, has language connectivity and activation, low myelin, and 55B is in the expected location relative to this sensory motor reference. But here's an atypical subject that's pointing toward this white dot here where the language connectivity and activity have shifted up. And the myelin, light myelin is actually up here, and the higher myelin is in the expected location of 55B. Here's an additional shifted subject, the same thing, where we've got language connectivity, language activity up here near this white dot instead of in the expected location. So that's one pattern, but turns out there's another pattern that we found where 55B actually gets split in half and the frontal eye fields join. So now we'll consider the eye fields, 55B, and the motor cortex here. And this is the typical relationship with the eye field split in half and a single 55B. And sometimes the eye fields fuse together and 55B gets split in half like this. So what does that look like in real data? We've got the same things as before, now adding the eye fields and motor cortex outlines. And we've replaced the sensory motor subregion with the dorsal stream visual functional network here. And we can see the I fields have connectivity to that, but 55B does not have connectivity to that. So here's a typical subject: language connectivity and activation, 55B, high myelin in the two I fields, but not in 55B, connectivity to the dorsal stream in the two I fields, but not 55B. But here's a split subject where we have two hotspots of language connectivity and task activation, with missing connectivity right down the center of 55B. And right in that same location, there's high myelin and connectivity to the dorsal stream. So the two I fields have come together here and split 55B in half. And note that all these different modalities are independent modalities are changing together. Here's another split subject. Again, lack of connectivity and activation right down the center of 55B, where we've got high myelin and dorsal stream connectivity splitting 55B in half. So we do have atypical parcellations, and it turns out that it's 11% of subjects have an atypical parcellation, whereas 89% have the typical topology. 4% of those are shifted and 6% are split. So we want our canonical parcellation based on the typical or population topology, but we still want to represent these atypical topologies. And even aerial feature-based surface registration is not going to be able to do that. So we need another way. Turns out we can use machine learning for this purpose. We can use a multi-layer perceptron to learn each area's aerial fingerprint in terms of its multimodal features. We'll make a few reasonable assumptions, such as assuming that V1 isn't outside the occipital lobe. We can simplify the problem doing binary classification for each area versus 3 centimeters in all directions across the surface. Combine across classifiers using the highest aerial probability across all 180 classifiers, and see what we get. So we can look at the outputs of the aerial classifier in a test-retest design, which we've got here at the individual subject level. So we've got a folding map, the group location of 55B, a myelin map, language connectivity. This is the raw output of the classifier and the final output. So this is a subject scanned and run through all the processing once, and here's the same subject scanned and run through the processing independently. We were able to find 55B in the expected location, but that wasn't really a hard problem. It's already lined up with the group location of 55B because this is a typical subject. But it turns out we have a split subject in the test-retest data set. We've got high myelin crossing the expected location of 55B in both the test and retest data. We've got a split in the language connectivity. We've got raw probabilities are split and the final probabilities are split, and that happens reproducibly across the test and retest data. Here's a shifted subject where we've got high myelin at the bottom of the expected location of 55B. The language connectivity is shifted up. We can see that the raw probabilities and final probabilities are also shifted up in this subject. So not only is the aerial classifier able to reproducibly identify area 55B in the typical case, these atypical topologies are stable in time. They're real neuroanatomy of these subjects, and the aerial classifier can pick them up. So a comparison of the detection rates across all of the areas. So we trained on the 210P group of subjects, and so it's not surprising that there's a high aerial detection rate of pretty much everything being yellow. But what is good to see is the held out data, the 210V data, also performs similarly. 96.6% of all cortical areas in all subjects are identified in this data set. If we leave out the task data, it works almost as well, 96.4%. So this means that the aerial classifier will work for studies without the HB specialized task fMRI battery, needing only T1, T2 field map and fMRI to use it. Note that's what I said at the outset. So what about reproducing the multimodal parcellation using only these aerial fingerprints? Well, we can apply the trained aerial classifier to both the 210P and 210V data sets to generate individual subject parcellations, and then average those parcellations across subjects to produce probabilistic areas. Here's probabilistic V1, M1, area 55B, and 46. And they're very similar across groups. These are tighter distributions, and areas like 55B with more variability are not quite as tight, or area 46, but very similar across the two groups. And so we can use these probabilistic maps to make maximum probability maps. Now we're displaying only the boundary gray ordinates again, like we saw earlier. Blue is for 210p, red is for 210v, and purple means that both had a boundary in that location. And you can see that this is mostly purple. So these two data sets are in very high agreement. The correlation, the parcellations is 0.965. And this is in line with those high correlations that we saw with the group maps originally. And this big focus on rigor and reproducibility now in brain imaging studies, and these are really highly reproducible parcellations. All right, to summarize, individuals have atypical layouts of cortical areas that we can't align with aerial feature-based registration. An aerial classifier can detect these areas and automatically replicate parcellation in individual subjects. The group parcellation can also be replicated, and these aerial classifier and individual subject parcellations are coming soon. So again, this is the full list of tools. Welcome your questions to the mailing list here. And again, I apologize for not being available in person to take your questions live. Thank you for your attention.